Okay, so we'll just dive right in. So the first thing we're going to talk about is protein structure, which is very important. I have to say most of the material in here is extremely high yield on the MCAT. So you should do well to know pretty much all of the information in this video, especially the things that are bolded on each slide. So a quick introduction to protein structure. Proteins can act as enzymes, hormones, receptors, transporters, antibodies, or a structural support both inside and outside of the cell. So we talked about the cytoskeleton and the extracellular matrix and some other videos, and you can see proteins are super diverse, and we're just going to discuss a little bit about that in a little bit. So proteins are polymeric chains formed by monomers known as amino acids, and there are 20 amino acids that you need to know for the MCAT, and we're going to discuss that on the next slide. So basically, amino acids interact three-dimensionally with each other to fold into a protein, and they're structured in 4 different levels. So we'll go over each one of them individually, and it's pretty important that you know all of them. But right before we do that, we're just going to discuss amino acids really quickly. So by definition, amino acids have a backbone that consists of a carbon molecule attached to an amino group, a carboxyl group, and a variable side chain. So if you look right over here, down by this little diagram, we have all the way to the left, that's that amino group. It's attached right over here to a carbon, which is known as the alpha carbon. So that is the carbon that's attached to everything else. We have the R group over here, a hydrogen on most of the amino acids except for proline. And then it's attached right over here to this carboxyl carbon, and we have this carboxylic acid group right over here. So this structure is the same for every amino acid. The only thing that's going to change is this side chain over here, this R group. And these differing R groups are actually what give each amino acid its specific properties. So in reality, there are actually hundreds of amino acids. Your body can modify different amino acids to use it for different things. Like hydroxyproline, for example, is a proline molecule with the hydroxyl group added to it. But you're only responsible really for the 20 proteinogenic amino acids. So those are the amino acids that we've been learning and reviewing, since high school. And if you look to the right, this diagram does a pretty good job at grouping them into 4 categories. Sometimes you'll see other categories like they'll have like sulfur containing groups, or they'll sort them into like ring structures, but these 4, I think this is adequate enough for you to learn. You do need to memorize this. I think that the best way to memorize the amino acids is probably flashcards. That's the way that I did it, and I think it was pretty effective. So you should know the name, the structure, the 3-letter code, and the 1-letter code for all 20 amino acids. It can only help you for the MCAT. And obviously, of course, you should know how each side chain interacts, with other side chains that are the same and other side chains that are different. Okay, so we're going to talk a little bit about the 4 structures. So the first structure is the primary level. So the primary structure is the simplest level of protein structure. It's simply the sequence of amino acids bonded together by peptide bonds. These are the only interactions that occur at this level. So if you look right over here to the right, that's exactly what it is. These little dots are just amino acids, these little circles, and the primary structure, we are just considered of the sequence and the order of the amino acids that are coming. The only interaction at this level are those peptide bonds. And actually peptide bonds are only present at this level. So all proteins have a primary structure, obviously, because the definition of a protein is that it's made up of amino acid monomers. So all proteins do have a primary structure. Secondary structure. So the secondary structure constitutes the initial and most basic level of folding. So this is when the protein actually starts to fold, and it arises from hydrogen bonding between backbone amino and carboxyl groups. So that's the important part to understand about secondary structure, is that the only, just like peptide bonds are the only sort of interaction that's going on at the primary level, at the secondary level, the only thing that's happening, the only sort of intermolecular interactions that are occurring is hydrogen bonding, and it's only between the backbone amino and carboxyl groups. So if you look to the right, you can see the 2 forms, the 2 motifs that are most common on the MCAT and are just super important to know for when you're learning biology and biochemistry. This one to the left is the alpha helix, and you could see it's a pretty good diagram, and it pretty much shows how the backbone, how those nitrogen and carbon, the carboxyl groups, are actually hydrogen bonding to form this alpha helix. And this is the beta pleated sheet, which is slightly different conformation. We're going to discuss both of these a little bit more in detail in just one second. But like I said, the most important part of the secondary structure is that hydrogen bonding occurs between the backbone. It does not have anything to do with the R groups just yet. Okay, so the alpha helices, beta pleated sheets. Alpha helices are often found in proteins that act as hormone receptors and ion channels in the cellular membrane. So those are those transmembrane proteins. You often find alpha helices in those proteins. This is because most of the hydrophilic interactions occur inside the helix, and most hydrophobic R groups point outward. So remember, when we're talking about the cellular membrane, it's a hydrophobic environment. So anything that's hydrophilic or electrically charged is not going to be able to associate into the membrane very well. So beta pleated sheets don't do a very good job of hiding their hydrophilic R groups. Alpha helices, however, just because of that coiled structure, they can orient things inward and outward toward the environment. So when we're in an aqueous environment, we want those hydrophobic R groups to be inside, and we want the hydrophilic groups to be outside. That's just the lowest energy conformation, and that's how these alpha helix proteins, these alpha helix motifs can stay inside the cellular membrane. One interesting fact is that alpha helices cannot contain proline. Proline is this molecule right over here to the right. So if you remember before, the basic structure of an amino acid, we have this alpha carbon right here. We have right over here the carboxyl carbon, this whole carboxyl group, and this alpha carbon is attached to a nitrogen over here. But if you notice, this is a secondary nitrogen as opposed to a tertiary nitrogen on the other amino acids. So that has some pretty big implications in how the primary structure affects the folding into the secondary structure. So because this is secondary, it causes a problem forming alpha helices for two reasons. So first, the secondary nitrogen caused by its unusual R group causes a kink in the peptide being formed. So because of this secondary nitrogen over here, it can't form sort of those straight long chains that the other amino acids can, just because of the actual geometry of that secondary nitrogen. So it causes a kink in it, which makes it very difficult to form certain motifs, especially the alpha helix. So the formation of a peptide bond with proline eliminates the only hydrogen attached to nitrogen, thus removing the potential for hydrogen bonding. So when we're talking about peptide bonds, this nitrogen is going to peptide bond with the carboxyl group of another amino acid. And the only way it can possibly do that is by getting rid of this H. So when we get rid of that H, there's no possible way that that nitrogen can hydrogen bond. And remember, when we're talking about secondary structures and alpha helices, it is the backbone interactions of the amino acid residues that are actually making that motif, that are creating the alpha helix. So that's just something to keep in mind. Proline is really just like a special case out of the 20 amino acids. It's actually referred to as an amino acid with an I. So in beta pleated sheet configurations, hydrogen bonding occurs between residues that are distant from each other and sometimes even on different chains. So compared to alpha helices, where it's just a couple of amino acids away and they're bonding and that's what's forming that coil, the beta pleated sheets are usually very far away. And sometimes they're even on different peptides. And that's what causes that sort of wrapping around, that long folding shape. And these sheets can have chains that run parallel or anti-parallel. So parallel means they're running in the same direction. That's usually two separate peptide chains. They're running in the same direction. And usually anti-parallel occurs with the same peptide chain because it's wrapping around, kind of like a snake doing a sort of meander. So every time it passes itself, it's going to be going in the opposite direction. Okay, so tertiary structure, which involves interactions between very distant residues. And when I say residues, I mean the amino acids that are bonded to each other. And it's important to understand that at the tertiary structure, all of these interactions are occurring between R groups, between those 20 different side chains. We're not talking about the backbone anymore. So there are 4 interactions that occur at this level that you must know. So we have hydrogen bonding, again, as long as the R group is allowing for hydrogen bonding, that can occur. Remember, it's R groups, it's not the backbone. We have van der Waals forces. They're sometimes known as nonpolar forces, hydrophobic interactions. We have disulfide bridges between cysteines, because remember, we have that sulfhydryl group that can bond to each other, and they can create disulfide bridges, which will stabilize the protein. And then we have electrostatic interactions. So we can have like polar interactions between charged side chains. We can have ionic interactions. So all 4 of these interactions are happening between side chains, and they all they all contribute to that tertiary folding of the structure. So the folding is actually driven by interactions between the side chains and the solvent as well. So this protein is going to fold. It aims to fold to adopt the lowest energy conformation state. So for example, like we mentioned before with the alpha helix, it's that same sort of hydrophilic pointing outward, hydrophobic pointing inward. That theme pretty much runs through secondary, tertiary, and quaternary structure. So hydrophilic R groups are going to be exposed outward, while hydrophobic groups will fold inward away from the aqueous environment. And if you look over here, this is an example of a tertiary structure. It's pretty simplified, but you can see the different bondings. So right over here to the bottom left, we have a disulfide bridge between two sulfur groups. This acidic and basic interaction over here would be electrostatic interactions. We got some hydrophobic interactions over by the orange, and then we have hydrophilic interactions over here. Okay, so quaternary structure is the largest structural level of some proteins, and it's important to keep in mind that not all proteins do have a quaternary structure. So they arise when two polypeptide subunits interact to form a multi-subunit complex. So if you look below, we have hemoglobin. I'm sure you're all familiar with hemoglobin. That would have a quaternary structure because we have 4 different subunits that are associating together, not with a peptide bond, but they're associating together and they're creating one larger complex. And we identify all 4 subunits as one complex known as hemoglobin. It's not an enzyme, but it is the same exact concept. So the forces that stabilize quaternary structures are the same as those at the tertiary level. So it's those interactions that we were talking about with the side chains in the last slide. And like I said, quaternary structures cannot be bonded by peptide bonds. They're 2 distinct polypeptide chains. So if there's a peptide bond between the 2 of them, then they would no longer be considered 2 distinct polypeptide chains, and the whole structure would just be considered tertiary because it's the same chain. Okay, so that brings us to the first review question. So read this over. I would pause the video, take about a minute to answer your question. And then when you unpause, I'm just going to go over the answers really quickly. So which of the following proteins does not have a quaternary structure? The correct answer is alpha tubulin. So let's go over it. A, hemoglobin is actually what we just discussed. It's actually made of 4 subunits that are not bound by peptide bonds. So that does have a quaternary structure. And the other 2, pyruvate dehydrogenase and ATP synthase, are 2 very complex proteins. And they definitely have multiple subunits. So basically, it's really just the absence or presence of subunits that we're looking for to identify if a protein has a quaternary structure. So alpha tubulin itself is a monomer of tubulin. So it's not a quaternary structure. Alpha tubulin itself is just a singular monomer, a single polypeptide chain. Once it polymerizes, that filament definitely does become a quaternary structure, but alpha tubulin at itself does not have a quaternary structure.